Okay, for the heart, everybody knows for the heart, you go to the cardiologist. For the hormones, you go to the endocrinologist. And for diffuse musculoskeletal pain, you go to the orthopedic. But nobody really talks about the lymphatic system, although it is a very important system. Oh, I have some nerve damage, I will go to the neurologist or the neurosurgeon. But nobody really talks about the lymphatic system. And it is a very important system. The lymphatic system is a supportive system, but by no means it is a useless system. It's a system that supports your immune system and your cardiovascular system. So we discussed in the past that the cardiovascular system is responsible for pumping the oxygenated blood. Excuse me? Okay. All right. So we have the blood being pumped to the tissue compartment by the heart. The blood being pumped to the tissue compartment by the heart. And we have some good oxygen-rich blood. And this oxygen-rich blood is poor in carbon dioxide and metabolic waste. And then we learned that the tissue is going to receive this oxygen. The tissue, please remember that the tissue is the target. You know, all these dots represent a nuclei. These boxes represent a cell. These boxes represent a cell. And this is a tissue. So we arrive at the tissue compartment. The tissue compartment. And we deliver from the heart we deliver to the heart the oxygen. Why does the heart pump oxygenated blood to the tissue compartment? For two processes known as aerobic respiration. And what's the second process? Oxidative. And obviously oxide refers to what? To oxygen. So oxidative phosphorylation. Oxidative phosphorylation. We have aerobic respiration and oxidative phosphorylation. Aerobic respiration and oxidative phosphorylation. Then obviously the tissue compartment is going to take the oxygen and then it's going to burn it, and then it's going to give us the waste, the carbon dioxide and the metabolic waste. We talked a lot about this, about how the car doesn't just take gas. The car doesn't just take gas from you go to the pump and then you pump gas, and then the gas is burned by the engine, and then you see all this fume and gas representing all of the waste. And then you see that there is a return of blue blood. There is a return of blue blood to the heart. This blue blood is rich in carbon dioxide. This blue blood is rich in metabolic waste. Now, we return back to the heart, and we've learned that the heart has two divisions. There is a right heart, and there is a left heart. There is a left, very big heart, and then there is a right, small heart. And both of them represent two pumps. Both of them represent two pumps. And this is a dividing septum. And then the deoxygenated blood is going to be pumped out to the lungs. It's going to be pumped out to the lungs. And the lungs are going to take the deoxygenated blood and return it back to the heart as freshly oxygenated, oxygen-rich blood that is poor in carbon dioxide. This is the cycle that we have learned about. Is this cycle clear to everyone? Anybody has any question about this cycle? What's the name of this blood vessel here who is going to be taking blood from the heart to deliver it to the tissue? Taking blood from the heart in order to deliver it to the tissue. This oxygen-rich blood that is going to the tissue. What's the name of this blood vessel right here? Please raise your hand. Anybody knows what's the name of this blood vessel here who's taking blood from the left heart? You should know that. This is the left heart going into the circulation to deliver to the tissue compartment oxygen-rich blood, poor in carbon dioxide for aerobic respiration and oxidative phosphorylation. Who can give me the complete name? The two parts. The complete name is two parts. What is the name of this blood vessel here? This here. What's the name of this blood vessel? Raise your hand, please, if you know. Raise your hand if you know the answer, please. Raise your hand. This one here, where the arrow is. This is the left heart. Blood is being pumped from the left heart to the tissue to deliver oxygen-rich blood, put in carbon dioxide for aerobic respiration and oxidative phosphorylation. Very clear. This one here. What's the name of that? Yes.
I think external cavity.
I'm sorry? Can you say it again?
I'm not sure. External cavity.
External carotid. No. The two descriptions. So that is a pulmonary. Pulmonary artery. That is wrong. Yes. So the aortic trunk is the name of the actual blood vessel, but I was trying to find out if this is an artery or a vein.
Artery.
This is an artery. So this is an artery. It's leaving the heart, so it's an artery. It's an artery that is carrying oxygen-rich and carbon dioxide for blood. What kind of artery is that? This is a systemic artery. Excellent. So now it's going to the tissue. Returning. Who can give me the two names for this blood vessels in terms of function? If I want to ask you about the aortic trunk or the external carotid, I would have a photo of the actual blood vessel like how it is in your lab practical 2 figures for next week. That one you will see the actual photo. Here we're talking generically about the anatomy and the physiology, the structure and the function. Excellent. So now we are returning back. So that's a vein. Very good. And we are returning carrying a high carbon dioxide partial pressure blood and a low partial pressure of oxygen. That's a vein. What kind of vein is that? This is a systemic vein. Excellent. Now, this blood vessel here, leaving the right heart, and going to the lungs. Leaving the right heart and going to the lungs. What's the name of that blood vessel here? Give it to me in 2 words. What is that? Excellent. Since it's going away from the heart, that's absolutely is a vein. Since it's leaving the heart, that is absolutely a vein. And since it is leaving the heart, it's going to the lungs. And since it's leaving the heart. It's leaving the heart. I got confused myself. It's leaving the heart. That's an artery. So, what kind of artery is that? Because it's going to the lungs. That's an example of a pulmonary artery. And the last thing is our friend here. What are the two descriptions of this blood vessel right here? Going one from the lung, heading two to the left atria, carrying inside this blood vessel is an oxygen-rich blood and putting carbon dioxide. What is that?
Pulmonary vein.
So that's absolutely is a vein, and it's coming from the lung, so that is a pulmonary vein. So you should absolutely have no problem identifying. If you're struggling to know the pulmonary artery, the systemic artery, that's a problem. It should be very obvious to you how to tell an artery from a vein. Artery always leaving, and then a vein is returning to the heart. Then the other part, systemic, if you're coming from the system or you're heading to the system. Pulmonary is if you're coming from the lungs or heading to the lungs. Now, where does our lymphatic system fall in this here? Well, our lymphatic system is a supportive system. They call it the green system. It's a supportive system. You see this green arc that I'm drawing here? In fact, it looks like this. You see this green vasculature here. What do you notice? Does the red side have a green supplementary lymph? This whole lymphatic vessel. Lymphatic vessel. Does the left side have a lymphatic vessel? Where is the lymphatic vessel only found? Where is the lymphatic vessel only found? Lymphatic vessel is only found where? Lymphatic vessel is only found where? Complementary and supportive and auxiliary to which side? Always the right side. The side that deals with waste. Because you see the right side is blue. Why is the right side in heart bluish? Because it's receiving bad blood and pumping bad blood to the lungs to be freshly deoxygenated. So that is our sewage system. That's our waste removal system. So the lymphatic system is a system that supports waste removal. So how does it support waste removal? Is the right heart not sufficient to pump out and remove the waste from the circulation? It is, but the issue is here in the capillaries. So now, let us zoom into the capillaries. You know when the blood gets to the tissue, when the blood arrives to the tissue, there's this compartment here. What's the name of this compartment? What is the name of this compartment? What's the name of this compartment here? This whole thing here. What do we call that? This whole thing. So this is a capillary. Very simple. That's a capillary. What happens in the capillary? What happens in the capillary is, you know, the blood is sequestered inside the blood vessels. The blood cannot come out unless it gets to the capillaries. So when you reach the capillaries, the arrow is pointing out, meaning that the red blood, the oxygenated blood, is pointing out, coming out from the capillary into the tissues. So the arrow is pointing out from the red half. The capillary has two halves. It has a red half, oxygen-rich half, and it has a blue, oxygen-poor half. True or false? True. So the arrow is coming out because we are trying to bring oxygen-rich blood to the capillary, and then the oxygen comes out into the tissue compartment. The tissue utilizes and burns the oxygen, and then all the ways point back. So now the arrow is pointing in the opposite direction. The arrow is returning. The waste, the blue waste is returning. This point is very clear. Does anybody get that? So all of the oxygen-rich components are flowing into the arteriolar side of the capillary, and all of the good stuff, the oxygen, the nutrients, the electrolytes are leaving through these pores, through these fenestrations, through these holes to be burned by the tissue, and then all the waste is returning from the tissue compartment to the blue part of the capillary. True or false? Is that part clear? Now, the question is, we learned that fluid does not move except from an area of a high, what? From a high concentration or high pressure. That's why we have a pump. Why do we have a heart? So it can pump with pressure. If your heart stops, why do you die? Because there is no pressure generator, so the blood is not moving. The circulation would stop. So the law says fluids will never move except from an area of a high pressure to an area of a low pressure. So fluids will move from an area of a high pressure down to an area of a low pressure. We discussed that. That's how the heart moves, how the blood moves from the heart to the tissue and returns back to the heart, to the right side of the heart, where the pressure is much lower. Is that clear? Now, if you see the arrow is pointing out from the arteriolar red half of the capillary that is rich in oxygen, the arrow is moving out. Where is the pressure higher? Is the pressure higher here in the capillary, or is the pressure higher here in the tissue? The pressure is higher where? Where is the pressure higher? Look at the law. The law says if the pressure is higher in one place and lower in the other place, then the movement will be from high to low. That's why in a waterfall, the pressure is highest at the top and the water slides down. So the blood moves from your heart, where there is high pressure, to your tissues, where there is low pressure. Now look here. The fluids are moving from your capillaries into the tissue. So where is pressure highest? Is it highest in the capillary or highest in the tissue? It is higher where? In the capillary. So the pressure is higher in the capillaries. That's why the fluid moves out from an area of a high pressure to an area of a low pressure. If you look at the other side, you will see that there is a pressure also in the tissues, and there is a pressure in the capillaries. Where do you think the pressure is higher? Is the pressure higher in the tissues, or is the pressure higher in the capillary? The pressure is much higher in the tissues. The pressure is much higher in the tissues and much lower in the capillaries. And that's why all the carbon dioxide, metabolic waste flows out from an area of a high pressure, the tissues, to an area of a low pressure, the capillaries. So, in your homework, there were many questions asking you, what is the name of this pressure here that is highest in the capillaries and lowest in the tissue and leads the blood to flow out. What's the name of that? What is the name of that pressure? Excellent. That's hydrostatic pressure. That is hydrostatic pressure. That's hydrostatic pressure. That's hydrostatic pressure. And the pressure that is guiding the return of the metabolic waste and the carbon dioxide back to the capillaries. What's the name of that? What's the name of that pressure? Is it still hydrostatic? It makes no sense. Hydrostatic means related to the heart, generated by the heart. Who's causing the metabolic waste? Who's causing the metabolic waste to return back to the bluish part of the circulation? Is it hydrostatic pressure? It's not. What's the name of that pressure? They talked about that in your homework also. What is the name of that? It's called the osmotic colloid pressure. The osmotic colloid pressure. That's where your lymphatic system comes in. Because the hydrostatic pressure is very efficient, 100% comes out. 100% of all of the oxygen and nutrients come out thanks to the hydrostatic pressure. But does 100% of the waste return back to the bluish part of the circulation? No, it doesn't. Because the colloid osmotic pressure is much weaker compared to the hydrostatic pressure. So how much actually goes back? 85%. So only 85% of the waste goes back. How much waste stays behind? 15. Who do you think pumps out this 15% of waste back to the heart? Who do you think pumps this leftover 15% negative metabolic waste that is building up in the tissue compartment that the colloid osmotic pressure could not return back to the circulation? The lymphatic system. That is the main job of your lymphatic system, to supplement and support your bluish part, your toxic part of the microcirculation that returns to the right heart, because it's not efficient. It only takes 85% and 15% stays behind. If this 15% stays behind, then some more is going to stay and pile up, and you're going to have a sedimentation of bluish toxic waste, and then the metabolic tissue is going to die. Is that clear? Any questions? So that's the main job of your lymphatic system. What's the other function of your lymphatic system? Ever heard of the B lymphocytes? The B lymphocytes? Anybody heard of the T lymphocytes? What are lymphocytes? Those are white blood cells that mature and develop where? In your lymphatic system. So if I ask you a question on the lab practical, name for me the functions of your lymphatic system. We just went over it. The first one is to remove all of the metabolic waste that stays behind, that couldn't be pumped by the colloid osmotic pressure back into the bluish part of the circulation, and now is being restored and brought back with this supplementary lymphatic system back to the right heart. What's the second job? The second job is it's the site for the development and maturation and growth of the B lymphocytes and the T lymphocytes to fight the pathogenic microbes and infection during the inflammation to combat all the infectious, pathogenic, all the infectious, pathogenic microbes. Infectious, pathogenic microbes. Infectious, pathogenic microbes. And the last job of your lymphatic system is to help in the digestion of fats. Fat. Lipid, fat digestion. So your lymphatic system has 3 jobs. Job number 1, we talked about it here, to return all the waste back to the right heart, not the left heart. Has nothing to do with the left heart, because left heart is oxygenated blood. So you have this lymphatic vessel that is transporting back all the waste. So we have B lymphocytes and T lymphocytes maturing to fight the microbe. That's job number 2. So that's job number 1, to restore all the metabolic waste. Job number 2 is the maturation of the B and the T lymphocytes. And job number 3 is the lipid and fat digestion. What does lipid and fat digestion mean? You see, if you eat a lipid or fat rich meal, eat steak, eat chicken, when your stomach breaks down that fat and then it moves, the fat is being digested and churned and broken down, and it goes into the intestine. Who helps with the absorption of the fat from your intestine into your blood? Those are lymphatic vessels known as lacteals. You need to know that. Lacteals. L-A-C-T-A-E-L-S. Please write that down. What are lacteals? Lacteals are lymphatic structures that absorb fats and lipids into the blood. Is that clear? That's the 3rd job of your lymphatic system. Job number 1, return all the waste back to the right heart. Job number 2, the development and maturation of the lymphocytes, B lymphocyte, T lymphocyte, for the infectious pathogenic microbes during the inflammation. And number 3, the lacteals, which are lymphatic structures that absorb fats and lipids into the blood during the digestion. Is that clear? Anybody have any question? Okay. Who can tell me the first function of your lymphatic system? I'm sorry? Removal of the waste and returning it back to the right heart. That's very important to be very precise. Number 2, what's the second job of the lymphatic system? Lipid and fat digestion. And we say digestion. Digestion is a big word. You know, they call the digestive system, but it's way more than digestive system. It's an ingestion system. It's a digestion system. It's a compaction system. It's the absorption system. It's the excretion system. Is the rectal canal not part of the digestive system? So they just name it digestive, but it's way more than digestive. So now what do the lacteals do? They help absorb fats from your intestine into the blood. And then the blood transports the lipid into the tissue, and the tissue break down lipid. We talked about sugar carbohydrate breakdown to get ATP from carbohydrates, and now we talk about lipid breakdown, get ATP from lipids. Any questions? All right. This is the main central concept that we will be talking to we'll be talking about in lecture today. So let's go over the PowerPoints, and we're going to keep returning and referring back to these concepts that we just discussed. Okay. Everybody, please go to the module section and download the PowerPoint for the lymphatic system, chapter 21. Okay. Alrighty, everybody's finished. Okay, erase this. Okay. There it is. All right. So, lymphatic system. So there are at least 10 times as many bacterial cells in a human body. This is why many students have the problem of assuming that all microbes are bad. No, we have two different groups of microbes. There are pathogenic microbes. Now those are bad. Those are parasites. Those are harmful. They gain advantage by causing you disadvantage. But there are friendly microbes. In fact, they're not just friendly, they are required. If you don't have them, you will have a problem. You may even die if you don't have those supportive, supplementary, helpful microbes. Such as, anybody heard of probiotics? What are probiotics? They help enrich and support these microbes who live in your, which system is the first, is your favorite system to be colonized and inhabited by the microbes? Is it the nervous system? Is it the blood system? Your blood system is immaculate. Your blood system is 100% sanitized. It doesn't have a single bacteria on it. Is it your muscular system, skeletal system, respiratory system? Which system is full of microbes?
GI.
Your GI. In fact, you have more microbes than cells. The number of microbes who live in your gastrointestinal system are more than the number of your bodily cells. And we like that. Why do we like that? Because they help with digestion. Some foods we eat, the enzymes we have are not strong enough to digest it. Number two, they fight for us. They fight other pathogenic microbes and they kill them when they enter our body. When you buy that sandwich from the deli infested with all of this nasty E. coli and infested with all of the, the deli guy is not thinking about your health and well-being. So it's gonna give you a sandwich that is full of disease. So when you eat that sandwich, those microbes are gonna kill all the bacteria that's in the deli sandwich. Why do they do that? Because they're territorial. We live here. We live in this person's GI tract. If you come in, we're not going to allow you to steal our place. So it's like a conflict of interest. They're not actually trying to help us. They're trying to help themselves, but in effect, we're getting help too. So long before your immune system starts to work and fight all of these microbes, the bacteria who colonize your GI tract, the one that you give probiotics to help them work. I had probiotic last night. Yummy. So make sure you have your yogurt. Yogurt with carrot and cucumber. Very good. Black peppers. Good God, amazing. You and the bacteria are happy. Everybody's happy if you eat that yogurt. So anyways, they are strong enough, and they're gonna kill any potential pathogenic infectious microbe. That's their first job. And they help with digestion too. They secrete some enzymes that is going to help us digest. But the potentially disease causing, what's their name? Pathogens. Some bacteria are pathogens, some are non-pathogenic. And the immune system is not an actual system. It's a collection of systems. It's like the IT department. They work with the network. You don't see them. They work behind closed doors. You don't get to see them, but they regulate all of the networking. So your immune system is a system that doesn't have like an actual conspicuous organ like the lung, the conspicuous organ of your most obvious organ of your respiratory system, the brain, the most conspicuous obvious organ of your nervous system, and so on. Especially concentrated in a true organ system, lymphatic system. So the network of organs, these are, you see, vein-like, because they are returning back to the heart. And they inspect for the disease, and they activate the immune response. That's known as the inflammation. We're going to talk about the inflammation today. So, what are the functions of the lymphatic system? Number one, fluid recovery. You remember that? Recovering the fluid. You see here, we say the fluid continually filtered from the blood capillaries into the tissue spaces, but the blood capillaries reabsorb how much? 85%. That means in the back, how much is lagging behind, how much is stuck behind is 15%. So of the water and about half of the plasma proteins enter the lymphatic system, and then they are returned back to the heart. That sounds like a lot practical question. How much percent is reabsorbed by the tissue capillaries? 85%. How much is reabsorbed and recovered by the lymphatic system? 15%. Number two, immunity. Remember we talked about the B and the T? Lymphocytes. So the excess filtered fluid picks up foreign cells and chemicals from the tissues and passes through the lymph nodes where the immune cells stand guard against any foreign matter. And that activates a protective immune response. Lipid reabsorption. You see that our friends are here, the lacteals. We talked about lacteals. Lacteals, they are found in a small intestine, and their job is to absorb dietary lipids that are not absorbed by the blood capillaries. Is that clear? That's the most important topic that we're going to discuss today. So, let's look at the lacteals. So if you look here, you have a red part of the circulation, and you have a blue part of the circulation, and then you have the lymph supplementing and supporting the bluish part of the microcirculation. So this is the small intestine as the microvilli in order to increase the surface area. Why do we have microvilli? In order to be able to increase the surface area. Now, the lymphatic system is similar to your circulatory system. The blood is flowing into blood vessels. Blood is flowing into blood vessels. The blood is flowing inside blood vessels. And the lymph is flowing inside lymph vessels. You see? They're similar. Blood is flowing inside blood vessels. Lymph is flowing inside lymph vessels. Now, what are the three layers of the blood vessels? The top layer, the middle layer, the inner layer. What's the name of the top layer? This is all lab practical questions.
External.
The tunica externa, and the middle one is
Media.
tunica media, and the innermost is the tunica interna. And guess what? The lymphatic vessels also have a tunica externa, the tunica media, and the tunica interna. Who's flowing inside blood vessels? Blood. Who's flowing inside lymph vessels? Lymph. What is the name of the cells that make? What's the name of the cells that make the blood vessels? They have a name. We talked about that last week. We said blood vessels are made of cells. That's why the cancer cell can make the blood vessels grow to give them nutrition. That's how if you break any of your rupture any blood vessels, a new blood vessel is made because it's made of cells. What's the name of the process is? Yeah, so angiogenesis is a process. Remember we said Genesis? Remember the first book of the Bible? The Genesis? How God made earth and the heavens and the waters, and Genesis is making. So the process is called angiogenesis, the making of blood vessels. I didn't ask about the process. I can ask about that on the lab practical next week. But I asked what's the name of the cells who perform angiogenesis. It's not angiocells. Somebody said it last time. We said that's a good guess, but that's not what it's called. Raise your hand, please. I cannot hear three people at the same time. Raise your hand. What's the name of the cell? Yes. The red blood cell is flowing inside the blood vessel. We're talking about the cell that makes the actual blood vessel. Yeah, very good. Excellent. Good job. Can you say it again? Endothelial cells. Those are the endothelial cells, the ones that have the capacity and the ability to regrow, provided that there are growth factors that stimulate the endothelial cell to grow. Lymphatic vessels transport the lymph. Lymphatic tissues are composed of aggregates of lymphocytes and macrophages that populate many organs in the body. Lymphatic organs, these are defense cells, are specially concentrated in these organs. They are separated from surrounding organs by connective tissue capsules. Lymph is a clear, colorless fluid. It is similar to plasma, but it is much less protein. They asked about that even on the NCLEX. They tell you, describe for me the difference between plasma and much less protein. Lymph is just like plasma, but it has more protein. So in another way, they're telling you that lymph is what? what did we say when you have a solution that is rich in proteins? It has a special name. Like the cytoplasm. The cytoplasm is a jelly solution that is rich in proteins. So is lymph. Similar to plasma, but it is much less. It doesn't have proteins. Because where the proteins are, they were in the tissue, right? So lymph is waste. Lymph is waste. Is carrying all the metabolic waste. Is protein waste? No, protein is something good. If you have protein in your lymph, that's a problem. Because the job of the lymph is to restore the 15% waste. Does it make sense to have protein in the waste? No. So it will be much less protein. The lymphatic capillaries, again, we tie back how we have blood vessels, lymphatic vessels, and blood capillaries and lymphatic capillaries. So you see, those lymphatic capillaries penetrate every tissue of the body, and they are absent from the cartilage, from the cornea, the bone, and the bone marrow. And the capillary wall is endothelial cells that are overlapping each other like roof shingles, and they are one-ended. They're closed on one end. And what do we see here? The endothelium creates a valve-like flaps. We will see now that the lymphatic vessels have something that distinguishes them from the blood vessels. You see, the blood vessels have valves where? On the inside, right? To prevent, if the blood is flowing up from your leg back to your heart, the valve is going to prevent it from refluxing and falling back. What's the name of the disease where the valves are defected and damaged? Where the valves are defected and damaged. What's the name? Varicose veins. There's blood pooling and blood vessels are broken and damaged. The valves for the lymphatic vessels, they're not in the middle. They are outside to regulate how much lymph coming in, how much lymph going out. Because the job of the lymphatic vessel is to receive waste and bring it back to the heart. Lacteals. What are lacteals? Again, we come to lacteals. Lacteals are lymphatic capillaries that are found in the small intestine. So there we go. That's the structure we drew earlier. You see now that the arteriolar side, you see red and blue. Remember we earlier drew that there are two halves of the microcirculation. One is bluish and the other one is red. You see that from the red side, the arrow is pointing in which direction? The arrow is pointing out from the blood vessel into the, what are these dotted circles? What are these dotted circles here? That's the tissue. So you see in the red side, the arrow is coming out from the blood vessel into the tissue compartment. But in the blue side, the arrow is coming out from the tissue into the lymphatic vessels, so that we return back to the right heart, to the bluish right heart. Now, do you see the valves? The valves are not in the middle. The valves for a blood vessel are now found in the middle. And you see now the lymph is supposed to enter through the flaps, and you will see that there are lymph. You see this lymph fluid is flowing inside the blood vessel. So you see, where is the lymph? The lymph is found where? Are you okay? So, you okay? Okay, so you see the valves are on the side. When it comes to the lymphatic vessels, the valves are on the side. But for the blood vessel, the valve is in the lumen to prevent reflux, otherwise we will have the disease varicose vein. I mean, that sounds like a perfect question on the lab practice. What's a varicose vein? What causes a varicose vein? Who is susceptible to having the disease varicose vein? Pregnant women, obesity patients, and occupational hazard, people who stand for too long. But for the lymphatic vessel, the valve is not in the middle. The valve is by the side to prevent the entry of the lymph fluid and the exit. Is that clear? So you see, the same way we have blood vessels, we have a lymphatic vessel. Tunica interna, tunica media, and tunica externa. So they converge. They convert into larger and larger vessels. So the collecting vessels course through many lymphatic nodes. There we go. 6 lymphatic trunks. They drain major portions of the body. It's jugular, subclavian, broncho, mediastinal, intercostal, intestinal, and lumbar trunks. There are 2 collecting ducts. There is the right lymphatic duct and the thoracic duct. This now is one of the most important key points in this lecture. Where do all the ducts empty their lymph? They empty it into the subclavian veins. They collect the lymph from both ducts, the right lymphatic duct and the thoracic duct. They will ask you this in clinicals. They will ask you this on the NCLEX. They will ask you this on the homework. They will ask you that on the quiz. They ask you that on the exam. This is one of the most important high yield concepts, that you have two huge lymphatic ducts. One is called the right lymphatic duct, and the other one is called the thoracic duct. The right lymphatic duct and the thoracic duct. Is that clear? The right lymphatic duct receives lymph from the right arm, the right side of the head, and the thorax, and empties into the right subclavian vein. Now you know why they call it the right lymphatic duct? That's a small duct. We're going to see its photo in a little bit. So the right lymphatic duct receives lymph from the right arm, right side of the head, and the thorax, and empties into the right subclavian vein. Now, if you look into the thoracic duct, that's a huge duct, larger and longer, begins at the prominent sac in the abdomen, known as the cisterna chyli. So, some questions ask you, the cisterna chyli is associated with the right lymphatic duct or the thoracic duct? Thoracic duct. And the right lymphatic duct empties in the right subclavian vein, and the thoracic duct empties into the left subclavian vein, and both of them empty into the subclavian. That's very, very important. So that's the photo we drew earlier. You see, now you have your red part of the microcirculation, and this is the association with the left heart, receiving oxygenated blood and pumping out oxygenated blood to the tissue. And you see hydrostatic pressure regulates the outflow of oxygen and nutrients, but the return is the problem. You see 85% returns through the bluish part of the microcirculation, and you see now there is a lymph outflow where you see the return, there is a return of all of the metabolic waste into the lymphatic vessels. So you see the lymphatic vessels are supporting the right-sided part of your microcirculation. And this figure here shows you what? Shows you the major prominent lymph nodes and lymph ducts. You see that's the right lymphatic duct and that's the thoracic duct, and both empty in which vein? Subclavian veins. So the right lymphatic duct empties into the right subclavian vein, and the thoracic duct empties into the left subclavian vein. When we talked about right heart failure and left heart failure, you need to understand what happens in right-sided heart failure and left-sided heart failure. What happens in right-sided heart failure? What happens in left-sided heart failure? Can you please say it?
The right heart failure is not working properly, so it causes systemic
Edema. So when the right heart is not working, that means the left heart is working, and the left heart pumps to the system, so there will be systemic edema. Excellent. When the left heart failure, left heart is not working, that means the right heart is working, and the right heart usually pumps to the lungs, so that means you will have pulmonary edema or edema in the lungs. Excellent. Very good, very good, very good. So that shows you the right lymphatic duct. That's the right subclavian vein. Those are the axillary nodes, and those are the lymphatics of the breast. You see, this is the drainage of the thorax, empty the thorax, and lymph flows under forces similar to those that govern the venous return, but there is no pump. So here we're not dealing with hydrostatic pressure. So the lymph flows at low pressure and slower speed than the venous blood. It is moved along by the rhythmic contractions of the lymphatic vessels. So we mentioned how blood is flowing in the microcirculation thanks to the effects of the hydrostatic pressure pumped by the heart. The heart. But the lymph, is lymph not a fluid? Lymph is a form of fluid. Lymph is a form of fluid, but this lymph fluid is moving from an area of a high pressure to an area of low pressure. So who's causing that pressure? It's not the hydrostatic pressure. Lymph flow at low pressure and slower speed than venous blood because it's moved along by rhythmic contraction of the lymphatic vessels. So those lymphatic vessels are pulsating. They are contracting constantly. The fluid is aided by the skeletal muscle pump. Those are causing contractions to propel the outer movement of the fluid into the lymph vessels. And there's an arterial pulsation where we have rhythmical squeezes of the lymphatic vessels. The thoracic pump aids the flow from the abdomen to the thoracic cavity, and valves prevent the backward flow, and they are rapidly flowing in the subclavian veins, drawing the lymph from a high concentration gradient to a low concentration gradient. So that's why exercise significantly increases the lymphatic return. Natural killer cells. Natural killer cells Those are special type of lymphatic vessels, lymphatic lymphocytes, cells that mature, grow, and develop in the lymphatic tissue. And they target cancer cells. Please remember that. They ask about it all the time. Which cells, which white blood cells, sometimes they want to be nice, so that they will tell you they will straightforwardly tell you that, listen, this is a lymphocyte that is known to target cancer cells. What is that? Those are the NK cells, natural killer cells. Large lymphocytes that attack and destroy bacteria, transplanted tissue, and host cells that are infected with viruses or that have turned cancerous. Sounds like a perfect lab practical question. Which lymphocyte is specialized in targeting cancer cells? And you say those are the natural killer cells. T lymphocytes. T lymphocytes, we said, remember when we discussed the involution? What are the two glands that undergo involution? The thymus and the pineal gland. T lymphocytes mature in the thymus, B lymphocytes mature in the bone marrow. So the activation causes proliferation and differentiation into the plasma cells. And the plasma cells produce antibodies. Who wants to explain for me what is proliferation and differentiation? What is proliferation and differentiation? Who could explain that? What is proliferation? What is differentiation?
Proliferation is not multiplying, and differentiation is not specializing.
They mean the same thing. Yep. So, proliferation and differentiation means you become a professional. So, B lymphocyte is immature. Like now you're a nursing student. Then, when you graduate, you're a professional nurse now. So now, that's why I told you, B lymphocytes, the activation of the B lymphocytes causes proliferation and differentiation into plasma cells. Those are the actual graduates who produce the antibodies. Do you get it? So, proliferation, differentiation means you've reached full potential and capacity and you can exert your function. So not every neuron in your brain was born as a neuron. No, it matured, it differentiated. The white blood cells, we talked about that in the blood lecture. We said that you have a stem cell that proliferate to become a neutrophil to target. What does neutrophil target? Bacteria. Eosinophils target viruses, and parasite. Eosinophils target parasites, and basophils target viruses. So that's an example of competency. That's an example of proliferation. So you must reach full potential after going through different successive stages, precursors, stages of growth and development. Macrophages. Back to see the macrophage. Macro means huge. Phage means eater. Those are the huge eaters because they swallow and they eat bacteria. Dendritic cells. What are dendritic cells? Those are branched, mobile, antigenic presenting cells. So those antigenic presenting cells, they fragment and rupture and break a piece of any invader. How do your B lymphocytes and T lymphocytes recognize that this is a bacteria that is pathogenic. This is a fungi. It's because of the antigen presenting cells. You will see this a lot. What does APC stand for? APC. APC is antigen presenting cells. Those antigen presenting cells fragment and break pieces of the pathogenic microbe and present it for other immune cells to degrade and destroy that bacteria. So what happens? What does the cop do? The cop arrest the criminal. But it is the detective or the judges, those are the ones who will completely get rid of this criminal and put them in the prison. Same thing, the white blood cells, those are antigen presenting. They're all about just arresting the bacteria and targeting the bacteria, identifying the bacteria. That's why they call them APCs, antigen presenting cells. Then comes the lymphocytes who will do the job of completely destroying and getting rid of these pathogenic bacteria. There are reticular cells. Remember you guys discussed reticular tissue in anatomy and physiology 1, A&P 1? Remember? You did epithelial tissue, you did muscular tissue, you did nervous tissue, you did different types of connective tissue. Reticular tissue, give me a particular organ that is completely made up of reticular tissue. Bone marrow, that's all reticular tissue. What else? What else besides bone marrow that is reticular tissue? Anybody know? Spleen. Spleen is completely made up of reticular tissue. Watch how I ask next week. Give me an example of an organ that is made up of reticular tissue. We just said spleen. Some people don't remember. So, if you're not going to pay attention in the lecture and you're going to be working on whatever on your laptop or sleeping, or then don't come in the end and say, well, I didn't know, I didn't. You have to pay attention to catch all of these points. Because you will be responsible for them, not just in this course, in the future also.
Before this, which one was the one you said? Bone marrow?
Bone marrow, yeah. Your bone marrow is completely reticular tissue, and your spleen is reticular tissue. Okay, so the lymphatic tissue is like a precinct, like a police precinct. So that's why do people get tonsillitis? What is tonsillitis? Is it inflammation of the tonsils? What are the tonsils? Tonsils are lymph nodes. So why do they get inflamed? Because they trap. They are like filter. You know how sometimes you filter through a strainer and some things get hauled up behind? Your strainer is full of now some debris that you don't want to be in your juice. Same thing. But those lymph nodes, they are a special type of strainers. They're a special type of filters. They are filters of white blood cells. So you have all these white blood cells waiting in line. It's like, have you ever drove your car and you're about to enter into the tunnel and you see like 500 cops like looking into who's passing by, who is this, screening? That's exactly what a lymph node is. It's an area where all the blood has no choice but to pass through, but there are so many white blood cells searching, filtering bacteria and pathogenic microbes and viruses and fungi and parasites. And once they find them, they trap them, and a big fight known as inflammation, we're going to talk about that today, takes place. So whenever the lymph node is inflamed, you know that they trapped a lot of pathogenic microbes and there's a big fight going on. Redness, pain, swelling, heat, loss of function because of the precinct is full of so much rebellion and activity and inflammation and combat between your white blood cells, immune system fighting those infectious microbes. Are we clear? So he shows you that's the lymphatic node. And the organs are made up of large aggregates. So we have primary lymphatic organs. We also have secondary lymphatic organs. Primary lymphatic organs, cells born and mature there. We just said the bone marrow and the thymus and the spleen, those are primary lymphatic organs. Secondary lymphatic organs, those are mature cells that are located in the lymph nodes, the tonsils, and the spleen, and those are immunocompetent cells that populate those tissues. Red bone marrow. The red bone marrow is involved in hemopoiesis, blood formation. We discussed that in the blood chapter 17. We said that red bone marrow is important for the differentiation of blood cells. Red blood cells and also the white blood cells. Separated from osseous tissue by the endosteum of the bone, and as blood cells mature, they push their way through the reticular endothelial cells to enter the sinus, and they flow away into the bloodstream. Now, if you look into the red bone marrow, you see the red bone marrow is highly invigorated with blood vessels that are bringing in oxygen-rich blood. And you know the thymus, that's a member of the endocrine, lymphatic, and immune systems, and they house developing lymphocytes like the T lymphocytes, and they secrete hormones regulating their activity. They are bilobed organs located in the superior mediastinum between the sternum and the aortic arch. Guess what comes again? Involution. This is the physiological degeneration of the thymus. Is that a disease? Is involution a disease? No, it's the natural, everybody's going to go through degeneration of the thymus. So this is the thymus, and you see that is the site of the maturation of what? What matures in the thymus? We mentioned that there is a particular structure a particular cell that develops in the thymus. What is it? Those are the T cells. That's why we call them T cells, because they mature in the thymus. And why do we call B cells B cells? They mature in the bone marrow. Lymph nodes. These are the most numerous lymph organs. And I mentioned a lymphatic reticular organ that is known as the graveyard for red blood cells. What's the name of that organ? Graveyard of red blood cells. The spleen. Spleen is the graveyard for all of the red blood cells. So those are the lymph nodes, and those are the ones who get inflamed. So everybody, look at the lymph node. This looks like a bean shape, like a kidney. But the kidney doesn't have those vessels, lymphatic vessels. Everybody look. You see a red vasculature and a bluish vasculature. What does that mean? Well, you have to know that this is a lymph node. So the blood has to come in through the red artery, and then all of the white blood cells are going to search it. It's like the TSA, when you have all your luggage has to go to be searched. So all the blood has to get inside and be searched, and then once it has been purified and all of the pathogenic microbes have been damaged and removed, then you see that all of the blood returns back to the efferent lymphatic vessels. Lacteals. You see that we have the small intestine, and you see those greenish structures here. What do they represent? Those greenish structures by your small intestine. What are those representing? Those are the lacteals. What is the job of the lacteals? Reabsorb the lacteals are important for reabsorbing the fats of lipids. So there are certain areas of major lymph nodes, like in the axilla and the inguinal lymph nodes. So now those are the diseases that target the lymph. Those are the diseases that target the lymph. The diseases are lymphadenitis and lymphadenopathy. What's the difference between lymphadenitis and lymphadenopathy? Lymphadenitis is swollen, painful node responding to the foreign antigen. This is swollen, painful node responding to a foreign antigen. Lymphadenopathy is a collective term for all lymph nodes and diseases. So lymphadenopathy is a generalized term for diseases that afflict the lymph nodes, while lymphadenitis, there is a swelling that is painful when there is a response to the foreign antigen. Now, what's the difference between invasion and metastasis? Invasion is when the cancer cell is locally spreading, invading neighboring tissue within the tissue compartment, having migrated through the lymph vessels or through the blood vessel to a distal location. But when the tumor flows from the breast to the bone, or from the kidney to the liver, or from the heart to the kidneys, from the liver to the kidney, that is going to a different distal site. This is called metastasis. So one of the prognosis, how we assign a stage for cancer, is this cancer stage 1, stage 2, is based on what? Did the cancer cell reach the lymph node? If the cancer cell reached the lymph node, then that's a major problem. If the cancer reached the lymph node, it's a major problem. Why? Because the lymphatic vessels are a way that, so if a criminal runs and gets to the subway, it's very hard to catch a criminal now. It's going to spread to a different location, to a different site. Same thing. We want to make sure that our cancer cell remains in the tissue. It doesn't spread to the bloodstream or the lymphatic nodes. So the cancer cells break free from the original tumor and travel to other side of the body and establish new tumors. The tonsils, those are patches of lymphatic tissue located at the entrance of the pharynx. They guard against ingested or inhaled pathogens, and this is known as tonsillitis. So tonsillitis is the inflammation of the tonsils, and it used to be removed. It used to be performed with the complete removal of these tonsils, and that's known as the tonsillectomy. So there are different types of tonsils: pharyngeal tonsils, palatine tonsils, lingual tonsil, but you know there are locations where the blood is going to be screened and searched for any pathogens. So the tonsils. We said that the spleen is the body's largest lymphatic organ, and the parenchyma of the spleen, parenchyma means what? Parenchyma, this is the major tissue, the functional tissue of an organ as distinguished from the stroma. There's a difference between parenchyma and stroma. Parenchyma is the original functional, structural and functional tissue of any organ. But stroma is a connective, supportive tissue. So the parenchyma of the spleen has a red pulp, you know from the color, red pulp. So that sinus is filled with red blood cells. That's why they call it red pulp. White pulp, that's the one with the white blood cells. Very simple. So lymphocytes, macrophages surrounding small branches of the splenic artery. That's the spleen. In innate immunity, we have pathogens. Those are agents capable of producing disease, including viruses, bacteria, and fungi. That's how we distinguish if the microbe is dangerous or not. Pathogens are agents that are capable of producing disease, including viruses, bacteria, and fungi. So there are 3 lines of defense against the pathogens. There's the first line of defense and second line of defense. A first line of defense would be your skin. Your skin is a first line of defense because it's a mechanical barrier. Your skin is like a fence. How do you protect your home from criminals? You have a structural barrier surrounding the home, don't you? In addition, your skin is not only a mechanical barrier, your skin is a chemical barrier. How is the skin performing structurally and functionally as chemical barrier? How? How can the skin act as chemical barrier? Do you know what is secreted on the surface of your skin? Dermacidins. What's a dermacidin? A dermacidin is a burning chemical. If any microbe touches it, it'll be burned. Your skin has what's known as an acid mantle. What's an acid mantle? An acid mantle is a thin film layer of acid. Acid mantle is a thin film layer of acid that once any pathogenic microbe touches it, it'll get burned. So that's our innate. Innate means natural immunity. So the first line of defense, that's your skin and mucous membrane. How can the mucous membrane act as first line of defense? You know mucus like glue? Glue. So you know how the mouse trap? The mouse sticks on a glue mouse trap? Your mucosa in your nose and respiratory tract and digestive system are like glue. They don't trap mouse. I hope not, it doesn't make it there. But it doesn't trap mouse. It traps bacteria. It traps fungi. It traps viruses. It traps different types of pathogenic microbes. And once it traps it, the white blood cells, the macrophages are gonna come, the neutrophils, the eosinophils, the basophils are gonna come and degrade it and kill it. Is that clear? So the second line of defense is when you have several innate defense mechanisms, and leukocytes and macrophages, those are antimicrobial proteins, natural killer cells, inflammation, and fever. This is the second line of defense. Yes. The second line of defense now, you thought your fence around your home is protecting you, but now the criminal jumps over the fence. You know, some of them do that. They don't need the door to be open. They jump over the door, they break the door. Some bacteria does that. They break through your skin, they break through the acid mantle. It is not a problem to them. Like some students, responders, and McGraw-Hill Connect is not a problem to them. They break off and they think nobody's watching. And then they get that letter and the video of them breaking the responders and the McGraw-Hill Connect. Oh, that part is shocking. Getting removed from the school because of a quiz. But anyways, some bacteria do that. It's not a problem to them. Your skin is not a problem. The acid is not a problem. They break through. So now, first line of defense is over. Now the second line of defense. Leukocytes and macrophages, antimicrobial proteins, natural killer cells, and inflammation and fever. This is your second line of defense. And we're getting closer and closer to the inflammation. Inflammation has 5 cardinal signs. Please write those down. The first sign is redness. Any inflamed tissue is always red. Redness, inflamed tissue has redness, swelling, redness, swelling. That's number 2, swelling. Inflamed tissue is red. Inflamed tissue is swelling. Inflamed tissue is hot, warm and hot. That's the third sign. Inflamed tissue is painful. Inflamed tissue is painful. And lastly, inflamed tissue will lose its function. And inflamed tissue will not do its job. If your knees are inflamed, can you bend your knee? So inflamed tissue is always suffering. Okay, the 5 are redness, swelling, heat or warmth, redness. You said redness, pain, swelling, heat, and loss of function. Okay? So, the third line of defense now is the adaptive immunity. So the white blood cells, like I told you, those are the cops. The cops are generalized. They call them for everything. Trespassing, check fraud, assault, thieves. No matter what, they call them for everything. But when the problem gets bigger, then the third line of defense comes in. The detectives and the judges, and that's for like robbery and homicide department, check fraud department, and all that kind of thing. Okay? Innate defense, they guard equally against the broad range of pathogens. They lack capacity to remember the pathogens. And there are 3 kinds of innate defenses: protective proteins, protective cells, protective processes. In adaptive immunity, the body must develop a tailor. It's not a one-size-fits-all. In the innate defenses, it's a one-size-fits-all. It's a generalized response to all the different microbes. But if you look at adaptive immunity, the body must develop separate immunity to each pathogen. Mast cells, macrophages, natural killer cells, dendritic cells, monocytes, neutrophils, basophils, eosinophils. Which of these are these cells here? First line of defense, or second line of defense, or third line of defense? Well, the third line of defense is the adaptive immunity, and those are the lymphocytes. But do you see lymphocytes here? No, they're not. So what do we see here? Those are the This is the second line of defense. Which cells in the second line of defense responsible for killing cancer cells? There's a lot of research involving these white blood cells.
That's the engine.
Yeah, the natural killer of the NK cells to make them wake up. Why are you not doing your job? You're supposed to be killing cancer cells. What's happening? So now they're trying to augment these natural killer cells. Okay, these are the neutrophils. So what happens? This is the project I was going to give you for the exam. So 60 questions, graded out of 50, so you have like 10 free bonus questions. But in order to do that, you have to do, I have a video summary of me going through these details here. This is very important. So this shows you here that this is the blood vessel, right? That's your blood vessel. And that's your tissue compartment. What's happening here? An injury, a laceration, a stab wound, a scratch. Those green cells are the bacteria, right? So the bacteria now enter. The door is open. They can't wait to get in. So they enter. When they enter, there are what's known as resident sentinel white blood cells. They call the radar. They call the circulating ones. So there are white blood cells waiting in line, looking, are there any microbes? If infectious microbes come in, they call who? They call the ones who are circulating. And the ones who are circulating, they perform chemotaxis. We know about chemotaxis? Chemotaxis, taxing. Chemotaxing is following the chemicals from the site of the injury. He said if you have an injury on your right pinky finger, why don't the white blood cells go to the left pinky finger? Because there are no chemotaxis, no chemicals being secreted here. And you see, this is the leukocyte squeezed between the cell, the capillary wall as they follow the chemical signals, and then you see the neutrophils releasing the cytotoxic. You see, now we have destroyed all of them. So this is a video that you have to sit down, watch me explain it, and make a summary. You can't afford to say no. You have to do this for your own success in order to pass this course. Because if you fail this course, now you gotta wait. This course is not offered. Now you have to wait. Is offered spring? Don't understand. Now you didn't take that with it. You gotta take that course with it. Yeah, trust me, this is a reality. So you can't afford to miss it. Bonus is a bonus, but you can't miss it. You do it. But this bonus, do it. That bonus, do it. Everything, participation, at every point matters. So that in the end, you pass, you leave, you get your degree, you succeed. Eosinophils, those are types of cops. So they deal generally. So they found especially in the mucous membrane. They guard against the parasites, the allergens, the allergy-causing pathogens, and they kill the worms. Okay? They are targeting the parasites, and they secrete hydrogen peroxide, and they promote the activation of basophils and mast cells. Okay, this is how the virus enters and replicates inside the cells. And you see now, you see those interferons, and those are perfect example of the chemicals secreted in the inflammation. The complement system represents a group of 30 or more globular proteins. This is a group of 30 or more globular proteins that work together. That's why they call them complement. The complement system, they complement each other, meaning that they work together to target the infectious microbe and to completely neutralize and kill that microbe. They engage with that microbe, synthesized mainly by the liver. They circulate in the blood in inactive form, and they are activated by the presence of the pathogen. So that shows the multiple activities of the complement system. So look at what the complement system does. First of all, the complement system identifies the target pathogenic microbe. It's like putting a sticker on it, putting a sticker to target it, and then it's going to lyse it. Lyse meaning break open, destroy it. Then oxidization, this is where we put a tag on a particular bacteria, and then the macrophage, whenever it sees that tag on you, is gonna kill you. That's how it works. What does, you need to know this. What does opsonization mean? Everybody write this down. What does opsonization mean? Opsonization is a process where you put a tag, you put a sticker. You see this green sticker? This green sticker was placed on the bacteria. The bacteria doesn't even know about it. The bacteria doesn't even know that they just put a tag on my back. But that's what your cells did. And then once the tag was placed on the back, what is this big yellow cell? This big yellow cell is a cell that is known as the macrophage to destroy. I'm sorry? Opsonization. Opsonization is a process by placing a tag on the surface. A tag is a protein, right? Proteinaceous tag, like antigen. Placing a proteinaceous tag on the surface of an infectious pathogenic microbe in order to kill and destroy that microbe. Okay. Then this process here, what is this cell? You see those cells here that are lining up your blood vessel? What's the name of those cells? Those cells here. What's the name of those cells? The whole thing is a blood vessel. Those cells are endothelial cells. Those endothelial cells, look carefully. What is being squeezed through them? You see this word here, extravasation? What does extravasation mean? Is when the white blood cell squeeze out through the endothelial cell and go in the tissue to kill the microbe. So the endothelial cells open up and make a gap. Clearance of immune complexes, those are immune complexes. So this is where now something similar to opsonization. But here you have immunoglobulin on the surface. So immunoglobulins attach together and stick to the surface of the particular microbe, and then the phagocyte is going to identify. So you see, phagocytes identify opsonization microbes, and phagocytes also identify immune complexes. This is the neutralization, the destruction of the bacteria, blocking of the ability of the virus to bind to the host cell, opsonization, promotion of phagocytosis, carried by macrophages and neutrophils, activation of complement system, and pore formation. These are the separate stages through which the complement activation takes place. There's a classical pathway, alternative pathway, and the lectin pathway. They dissociate into different components, and those different components, they arrive to this C3A and C3B. C3A gives us inflammation, and C3B products lead to good clearance, phagocytosis, and cytolysis. This donut, what is membrane attack complex? Please remember that. Membrane attack complex, or the MAC, M-A-C. What is the MAC? What is the membrane attack complex? It's when you put a donut. The donut is known as to have what in the middle? There's a hole. Why are we trying to put a donut on the surface? Why are we trying to place a donut on the surface of a cell? Why are we trying to place a donut on the surface of a cell? So the water can go in and inflame, and burst, explode the bacteria. This is, we have different tactics to kill the pathogenic microbes. Put a tag on it, put an immunocomplex on it, and put a hole on it, donut hole on it. Because when you put a hole on the surface of the microbe, the water flows through the hole and the microbe it becomes full of water and then bursts. So what does the complement system, you see? C9, C9, C8, C7, C6, those are complement. So the complement protein make the MAC, the membrane attack complex, make this donut. Natural killer cells continually patrol the body looking for pathogens and disease host cells. The natural killer cells attack and destroy bacteria, transplant cells, cells infected with viruses, and cancer cells. The natural killer cells, they release what's known as perforin. Please look at the name. Perforin. What kind of thing would that be? Perforin. Exactly, perforate. So they make a donut too. So how do natural killer cells kill their enemies? The natural killer cell releases perforins, meaning proteins that make holes, which polymerize and form a hole in the enemy cell membrane. That's the first stage. That's the enemy cell. The big cell is the enemy cell. The natural killer cell is a small cell. Then what's known as granzymes. Granzymes from the natural killer cells. What are granzymes? Granzymes are enzymes. You see these dots, granzymes? What goes through the granzyme is not water. In the MAC, the complement system, make a hole, the water goes through it, explode. But here, the natural killer cells make perforin, but does water go through? No. Granzymes secreted by who? By the natural killer cell itself. Granzymes go through, and then granzymes from the natural killer cell enter the perforin hole and degrade the enemy cell enzymes. And now the enemy cell enzymes are destroyed. And then the bacterial cell is dead. When the bacterial cell is dead, who comes? Macrophage. What does macrophage mean? Macro means big, phage means eater. So they come and they swallow and they eat the destroyed cell. Fever. Fever is an abnormal elevation of the body temperature. So the thermostat in your brain increases. So the synonym is known as pyrexia. So fever is always a result of trauma, infection, drug reaction, brain tumors, and it's an adaptive defense response. And it does actually more good than the harm, because it heats up the body. It warms the body so it can fry all of the microbes. And it elevates the metabolic rate and accelerates the tissue repair and inhibits reproduction of bacteria and viruses. Inhibits reproduction of bacteria and viruses. You see, this is the course of a fever. What happens in the course of a fever? You see that usually is the normal body temperature, and then you see that the hypothalamic, so in the hypothalamus, there's a hypothalamic nucleus known as the thermal nucleus of the hypothalamus. That one is responsible for a temperature, like a thermostat. Increase the temperature, and then the temperature rises. But you know, if you have fever, what are you supposed to do? Take a shower. It'll go right away. If you ever have a fever, go in a shower, particularly cold shower. I know you can't handle it, but if you do it, oh, you will thank me. Thank God, actually, but yeah, that's what happens. Take a cold shower, the fever is gone. Don't give the baby who has a fever cold shower. It's too much. Give them like a normal, not warm, not hot water, but give them like not a cold though, not cold. You can handle cold water. If you have a fever, go and get, you know, the ice bucket challenge. Oh, you do that one. If you have a fever, you do that one. Please believe me, You will look for the fever where it's gone. It's gone. I wish I didn't call out sick. Yeah, actually that's very true. I did it one time. I had such a bad fever. I was shivering. I can barely walk. So I got into the bathtub and I turned the cold water, and once the first layer just hit me, and then the fever was gone. I was refreshed. Then I was like, what are we having for breakfast? Before, I was not in the mood to eat. I was just sad. I was reflecting on my old days, I'm gonna die. But now I'm looking for breakfast. So now, take a cold shower, it will take the fever right out. But the fever is good, but you have to have a cold pad and a fan. You know the fan? If you have a baby, especially. All those parents, their baby has a fever, they can't wait to give Tylenol, and they can't wait to give the acetaminophen. That will stop more fever from developing. What about the old fever? Who's gonna make it go away? The cold pad. Because when the brain is hot, you're gonna fry the brain, they're gonna have a seizure. These parents come into the clinic. Oh, my daughter, she has a fever and she had a febrile convulsion. Well, you're the cause of it. Because you didn't put the cold pad. Well, I gave her the medication. The medication is going to stop additional fever. But the old fever is still there. You're going to put the cold pad, not very cold for the baby. For you it's okay, but not for the baby. For the baby, you put like normal water, just the water from the faucet. You put it, then the water is going to absorb the heat from the brain of the child, and then it's going to remove. And then the fan, keep the fan rotating to cool off the head. That's the brain. You can't afford to hurt your brain. And then you're going to cool, and then the baby will be okay. Okay? These doctors, they just can't wait to send you to the pharmacy. Their friend, by the way, he's their friend. So they send you to the pharmacist, and he gives them the commotion. Shame on all of them. But don't forget, use your cold pad and your cold showers. You wouldn't even need a medication. My mom used to use, what's the name of that thing? Monjaro. Now she lost her vision in the right eye. Now she doesn't take any medication. She said all of them are bad. So now she just uses like herbs. And it's actually working. So she has diabetes. So she walks in the morning, in the very morning, she walks fast. And she had nasal congestion. Like, Mom, take that fluticasone propionate nasal spray. She said no. She said she was sniffing this plant. And actually that plant was, my grandma did too. She put it in fire and the smoke came out of the plant. And she was like, sniff this. And I was like, no, fluticasone. She was like, no, just try it. And I swear, I just took one sniff. It went through my brain, and like all the congestion was gone. So I'm restoring my faith in herbal medicine. So yeah, my mom stopped all her medications, blood pressure, diabetes. She's just relying on her exercise and plants and herbs. Yeah. This is what we repeatedly talked about. You puncture a wound, and all of the infectious microbes come in, and you have two compartments: the tissue that is infected with the pathogens and the blood vessel. We talked about that. Those endothelial blood cells, they're gonna widen up. They're gonna make gaps. And who squeeze through the gaps? The white blood cells. And they're gonna go and they're gonna fight all the bacteria. I talked to you about this with chemotaxis. You see, the white blood cells in the tissue are going to call the white blood cells circulating in the blood vessel. They're not going to call them by phone, by sound. They're going to call them with chemicals. And then they're going to squeeze. This process called diapedesis. I still remember my professor when I was in college saying, I love this word, diapedesis. Diapedesis means when the white blood cells squeeze through the holes of the blood vessels.
Isn't that extravasation?
Extravasation, absolutely.
It's the same.
It's the same. Okay. Extravasation means that you exit the blood vessel from the blood vessel into the tissue compartment. But diatheresis is the ability of the white blood cell to squeeze out anywhere. It can go from a blood vessel to the tissue. It can go from, it's just describing the ability of the endothelial cell to cooperate with the white blood cell, like opening up endothelial gap to allow the squeezing.
So first is diatheresis and then it's extravasation.
Yeah, so extravasation is the complete exit through the process of diatheresis. And you know what chemotaxis is? See what my mouse is? Chemotaxis is when the white blood cells taxing to the site where the chemicals are being secreted, meaning the site of the inflammation, where there is redness and pain and swelling and heat and loss of function. Okay, this here shows us now the lineage of white blood cell. There are two lineages. There's a lymphoid progenitor, and then there's also what's known as a myeloid progenitor. A myeloid progenitor, and you see that. So we're gonna have a break at 4:45 because we're gonna have a break till 5:30. Okay? So it was supposed to be from 4 to 4:30. I'll give you from 4:45 to 5:30. Go and eat everything. and enjoy the beverages and even take a nap. And everything you do, then we meet in the lab at 5:30. Okay, those are two lineages, common lymphoid progenitor and common myeloid progenitor. Okay, we have, please remember, I talked about this in the blood lecture, and that we have thrombocytes. Thrombocytes are also known as what? Platelets. Platelets come from the mega, the huge. Look here, where my mouse is. They will ask you that. I promise you they will ask that on the NCLEX. They're going to tell you either the thrombocytes come from who, you say the megakaryocytes. Or they will ask you, the megakaryocytes give rise to what? You say the thrombocytes. They love that. This one, megakaryocytes. And also they ask, the monocytes give rise to macrophages. Or macrophages arise from who? You say the monocytes. Very important. Basophils, the neutrophils, and the eosinophils. This is the Y-shaped antibody. This is the Y-shaped, this is the Y-shaped antibody. The Y-shaped antibody has what's known as variable regions and what's known as those variable regions are the regions where the antigen is going to bind to. How does the antibody, remember when we said the A blood type will have anti-B antigens? So how do the antibodies of the A blood type, the anti-B, recognize B blood? With those antibody binding sites in the variable region. Okay, those are different 5 classes of antibodies or immunoglobulins. Immunoglobulin G antibody accounts for 80% of all the antibodies. Immunoglobulin E attaches as an individual molecule to the exposed surface of the basophil. Immunoglobulin D is an individual molecule on the surface of the B cell. Immunoglobulin M is the first class of antibodies secreted after the antigen is encountered. Immunoglobulin A is found primarily in glandular secretions such as mucus. Okay, this is the life history of the B and T cell. Did we not talk about this? Did we not say that the bone marrow gives rise to all blood cells, but B cells mature in the bone marrow, but T cells mature in the thymus. Then B cells differentiate to become plasma cells. Remember, megakaryocytes give rise to who? Platelets. Monocytes give rise to who? Macrophages. And B cells, when they get activated, they give rise to who? Plasma cells. And plasma cells give rise to who? Antibodies, the Y-shaped antibodies. Okay, so that here shows us a bunch of videos that show you can watch them at your leisure. We discussed that. The B cells and the T cells, okay. Antigen presenting cells, we discussed them. Those are, remember we were talking about the APCs? The APCs are antigen presenting cells. They encounter an antigen, they internalize it by endocytosis, they digest it into molecular fragments, and they display the relevant fragments in the grooves of the major histocompatibility proteins. Antigen presentation. So the wandering T cells inspect the antigen presenting cells, and the antigen presenting cell only displays self antigens, the T cell will disregard it. But if it's presenting non-self, it will initiate an immune attack. Antigen presenting cells alert the immune system to the presence of a foreign antigen. Okay. So display relevant fragments in the grooves of the main histocompatibility complex. See, that's the, let's look at how APC cells work, antigen presenting cells. Phagocytosis of this green pathogen first. Number two, the lysosome is going to fuse with the lysosome is going to fuse with this phagocyte, and then the antigen and the enzyme will mix to have a phagolysosome, and then the antigen is degraded. This antigen residue is voided by a process known as exocytosis. This residue, before it's exocytosed, a fragment of it, the processed antigen fragment known as the epitope. Please remember that, the epitope. What's the epitope? The epitope is a fragment of the processed antigen fragment. It's going to be placed at the MHC protein, and that's how we present. See why they call them antigen presenting? Because they present a fragment of the processed antigen. They present it at the surface, meaning, hey, we just chewed inside of us some really dangerous microbe. So now the T cell is going to come, they're going to find the epitope. The epitope is the one who's going to guide the T cell. Cellular mediated immunity is a form of specific defense in which the T lymphocytes directly attack and destroy disease or foreign cells. The immune system remembers the antigens and prevents them from causing disease in the future. There are four classes of these T cells, known as cytotoxic T cells, helper T cells, regulatory T cells, memory T cells. Now, the first step is the T cell is going to bind to an antigen presenting cell. Remember the epitope. So the T cell binds to the epitope fragment. The second step is known as the co-stimulation, where the T cell binds to a second protein on the antigen presenting cell. Then, whenever the T cell binds to the second protein, this is known as the co-stimulation protein. Once the T cell is now bound to the epitope and the co-stimulation protein, then there will be a degranulation. There will be a clonal selection, where T cells undergo repeated mitosis and they produce a large number of effector cells and memory T cells. Those are called effector cells. They attack. Those are effector T cells. They attack and destroy abnormal cells with the lethal hit, and those helper T cells secrete interleukins and stimulate multiple forms of the attack. Interleukin secretion is when the activity of natural killer cells, B cells, or cytotoxic T cells that's leads to the development of the memory cells. So memory cells means what? You remember this pathogenic microbe if it comes again. So what's the role of the helper T cell? The helper T cell, here you have the helper T cell secretes. You see they have a green color. This is an excellent demonstration because it tells you the three things that the helper T cell does. It secretes green proteins, purple proteins, orange proteins. This is not the color they secrete. This is just to help you understand. That's a model, just a model. But it does three things. It secretes some substance that would promote macrophage activating factor and macrophage activity leukocyte chemotaxis inflammation. This is a non-specific defense. We recruit cytokines is for like chemotaxis to recruit white blood cells. Number two, secret interleukins. Interleukins promote clonal selection, known as humoral immunity. Please remember that. Clonal selection of antibody, that's known as humoral immunity. And lastly, the helper T cells, they secrete interleukins that would promote clonal selection of the cytotoxic T cells. That's known as cellular immunity. So they're really helpful cells. And that shows you a dying cancer cell is being attacked by those helper and by these cytotoxic T cells. The immune system follows primary responses in cellular immunity, and following the clonal selection, some cytotoxic T cells and helper T cells become memory cells. The T cells recall exposure. So upon re-exposure to the same pathogen later in life, memory cells launch a quick attack so that no noticeable illness occurs. So the person is immune to the disease. That's the B cell and that's the plasma cell. Humoral attack. You have antibodies, have four mechanisms of attack against antigen. Neutralization, complement fixation, agglutination, we discussed agglutination, and precipitation is the culmination and the end of the process. Neutralization is where you have antibodies that mask pathogenic region of the antigen, and complement fixation is when IgM or IgG bind to antigen, change the shape, and initiate the complement activation, which leads to inflammation, phagocytosis, immune clearance, or cytolysis. The primary defense against the foreign cells, bacteria, and mismatched red blood cells. You see the agglutination? That's the agglutination where we hook up together the cells that possess the wrong or the non-self antigen. This is the primary response. So immune reaction is brought about by fixed exposure to an antigen. Okay, now if you look at the humoral immunity responses, usually if you have a particular infection, everybody, oops, okay, what is this figure trying to tell you? This figure is showing you a very important concept. That let's say you had a first exposure. You just got the disease for the first time. You've just had a first exposure to a foreign antigen. And then what happens when you have a re-exposure to the same antigen again? So let's look at the differences. In the beginning, you have rising levels of immunoglobulin M up to the 10th day. And after the 10th day, the IgM levels will subside. And the IgG levels will start to rise after the 5th day. And they will extend up to the 20th day, and then they will return. That's when the first time you encountered the foreign pathogenic antigen. Now, let's look at what happens when you become infected again with the same antigen. Look at what happens. Who skyrockets to the roof? Is immunoglobulin. So that's the memory. That's the memory immunoglobulin. So whenever you're asked which antibody, antibody means immunoglobulin, same thing. Which immunoglobulin, if you do blood work and you find skyrocketed amounts of immunoglobulin G, it tells you that this patient is having a second re-exposure to this particular antigen. Now, this boy here is known as the boy in the bubble. The boy in the bubble is a child that was born with a genetic disease, and there is an immunocompromise. So every single second, you're dealing with different types of infectious pathogens entering your body. You don't even feel it because your immune system, thank God, is competent enough to destroy them. But this baby here is suffering from a disease that their immune system is inactive. So they had to live in a sterilized bubble. This bubble will only bring in oxygen and remove carbon dioxide, and the baby cannot exit. So they lived in there up until, I think, 12 or 11 years old, and then the child died. This was a case of severe combined immunodeficiency disease. So that shows you your immune system is working in the background, but you can appreciate it. It's saving you from millions of diseases by the minute. And this child had this, what's the name of the disease? Severe combined immunodeficiency. Now, if you look at the HIV virus, the HIV virus is an RNA virus. So there is two R's about, there is three R's about the HIV virus. The first R is the RNA virus. The second R is a retrovirus. The third R is a reverse transcriptase virus. What does that mean? RNA virus, meaning that it is nucleic acid, is not DNA. There is DNA viruses, and there's also RNA viruses. The HIV virus has an RNA virus, just like the coronavirus. Then, moving on to the next stage. It's a retrovirus. What does a retro mean? Because it's like back in the day, from back in the day. Actually, retro, back in the day, HIV used to be the #2 killer in the whole world. It killed so many people, especially the people who are disadvantaged and the people who are living in poor places. It killed the vast majority of them. The rich people, not that many. And then, or the educated or the intellectual, didn't kill that much. And then you realize that it killed poor and homosexual men. Homosexual male, the first one to have the HIV virus. It killed so many of them. And then the homosexual men who went share the needle of the drug with other men who are heterosexual or with other women, and then the disease is spread out to the general population like that. And it eradicated so many people, especially in the poor, impoverished areas and countries in the world. And now they got to learn how the virus works. It's the RNA virus, that's the first R. #2 is the retrovirus, because it converts the RNA retroactively into DNA. That's why they call it retro, because it has RNA, so it has an enzyme that retroactively converts the RNA into DNA. What's the name of that enzyme that enables the HIV virus to convert its RNA into DNA? It's called reverse transcriptase. That's why we say 3 R's. HIV virus is an RNA virus, is a retrovirus that uses reverse transcriptase. Any questions? Are there any questions? Okay, the Kaposi sarcoma is the #1 most conspicuous sign of HIV. The patient has Kaposi sarcoma. Those are the purplish, bluish lesions that appear around the torso or the neck. You know, anybody heard of the movie Philadelphia by Tom Hanks? He got the Oscar. Yeah, it was about the Kaposi sarcoma. And you know, he was fired from his job because he had AIDS. And you know, he sued his company and everything. So yeah, so I learned about it long before I knew about it because of the movie, because he had the Kaposi sarcoma, and they were like, "What is that? You have virus? You can't work here." And you know, it's against the law to fire somebody because of their disease. If the disease is not contagious, but the people didn't know it was not contagious. It is contagious by sexual activity or exchange of sexual fluids and intravenous, but it's not like cough. People didn't know. And some people still don't trust that. They believe that, you know, if you have HIV, it may spread by aerosol. Not sure. If the virus touches air, it will die immediately. So the HIV, the virus is dead. The virus is dead because life for us, when you say life, for me, I'm a Muslim, life means the soul. God put the soul in you, S-O-U-L. Christians believe the Holy Ghost, the soul. In science, they don't believe that. In science, they don't believe soul. So they can't even define what life is, but they say life. We can't define it, but we can define the signs of life. Meaning homeostasis is one of the signs of life. If you are alive, you can do homeostasis, right? If it's hot, you're gonna sweat. If it's cold, you're gonna shiver. Can a dead body sweat and shiver? So it's dead and you're alive. So we can't define life, but we can define the signs of life. Another sign of life is metabolism. You can metabolize food. Can a dead body metabolize food? No. Stuff like that. Reproduction. You can reproduce, have a baby. Can a dead body have a baby? No. So they cannot define what life is, but they can know the signs of responsiveness. You can respond. But they say, you know, they found a homeless person with a drug abuse or overdose on intravenous injection, and they realized that this person is unresponsive, meaning that dead. So the virus is, according to science, dead because it cannot perform metabolism, it cannot perform homeostasis, it cannot perform reproduction. It needs you, it needs your cells to hijack your machinery, to hijack your enzymes, to hijack your ribosome to make its proteins to survive. Okay, everybody knows about this. HIV transmitted through blood, vaginal seminal secretion, blood, breast milk, sexual intercourse, contaminated blood products, needles, not transmitted by casual contact, and undamaged latex condoms is an effective barrier to HIV. Any questions? Okay, let's take the attendance before we go to Zuleika, Tanae. I remember now. Teen. You know teen? Teen in my language it means, you know the figs?
No.
You don't know fig?
No.
You don't know figs? You know what figs are?
All the fruits.
Figs and olives, if you eat figs and olives, I'm telling you a secret, guys.
No, yes.
Sure, figs and olives is very excellent. Mix figs with olives and eat them. I'm not gonna tell you, you'll thank me. Okay, Shara. Next one. And if you're pregnant, you wanna have a baby, you wanna have a smooth birth, you don't wanna bleed endlessly, you don't wanna feel the pain, dates. Eat dates. And the dates, and actually the dates you're gonna give birth, you're gonna get rid of it, you're gonna get done with it quickly. 9 months, people gotta wait 9 months, you, 7.5. Ruby is gone. and no pain. He's gonna be your favorite child. He gave me no pain, I'll never forget it. All of you guys gave me pain. Yeah. You guys said it to the sibling. I said to my mom, I didn't give you pain when I came out, did I? You gave me the most pain. I thought I was the easiest. Lorna.
Here.
Brianna Harborough.
Here.
Jean. Jean. Yeah. Manfred. Diana Fanous. Brianna.
Here.
Zeta.
Here.
Sean. Deshane. Natasha. Vanessa. Samantha. Okay, so I'll give you one last thing. Oh, I didn't forget to turn it. Okay, so this